Greetings everyone, I am Huzaif and my topic is agrigenomics. My domain is agrigenomics. My prompt statement is genomic prediction of oryza sativa hide. Agrigenomics is a significant field that concentrates on the genetic analysis of plants by utilizing genomic technologies. My goal is to predict phenotype traits using genotype data. My first objective is to build a GWAS file mapping genotypic data to phenotypic data, as we know that phenotypic traits are affected by the genotypic data. To build a phylogeny tree, my other object is to build a binary tree for the oryza sativa subpopulations, as phylogeny tree helps us to find the common ancestry, and while using the common ancestry we can find which two plants fall under one cluster or fall under one group of plants which are closer in the evolutionary timeline. Third objective is to classify the sample using machine learning algorithms. Third is to predict the GEBV height of oryza sativa. This is genomic observation. Height of oryza sativa using machine learning techniques. Third is to build a GUI application which will be a website using React and Flask. So genotypic data refers to the genetic makeup of an organism, including all the genes and all. Genotypic data are like the eye color and all. Whereas in the phenotypic data, it refers to the observable traits of an organism like height, weight and eye color. This eye color, whether it be a green or blue, that depends upon the genotype of it. So phenotypes, the effect of the phenotypes, genotypes are a factor which affects the phenotypic data. Then for the data collection, we'll be using the SR4R data, which is online available on the WARMAP RICE website. We'll be downloading 3 files, which will be cultivar information file for getting the cultivar IDs and the major plants. Third will be genotypic information of the file using the variation IDs, which I use 22 variation IDs in this, and phenotypic information file to find out the height. And then we'll be mapping the genotypic data to the phenotypic data. So first, the data collection has been done. As you can see, the various 22 IDs, 22 chromosomes have been taken into consideration. Third, then we merge the plant height data with the chromosomes, which will be merging of the phenotypic data with the genotypic data. Then the data pre-processing done, like replacing the missing values, finding the sequence, mapping genotype with the phenotype correctly, also taking out all the outliers in the data set. Then the second objective for us is to align the sequence to common ancestor using algorithms like UPGMA, Smith-Waterman and Needleman. These UPGMA algorithm is used for phylogenetic tree, and Smith-Waterman and Needleman ones is used for multi-sequence alignment. Multi-sequence alignment is a tool to identify functional domains and conserved regions, evolutionary relationships and common patterns between genes. There are two types of multi-sequence alignments. One is the progressive alignments and second is the iterative alignments. We'll be using the progressive alignments for this project. Tools for MSA. There are many tools for MSA. The 4 tools which you know are Cluster W, Cluster Omega, Muscle and T-Coffee. In this project, I use Cluster Omega, Cluster W and Muscle, and I found that Muscle is better than other two, so we'll be using Muscle in this. So there are 2 methods, and also for the making the phylogenetic tree. One is methods. There are 2 methods, distance based and characteristic based. Among them there are another 2 methods, which is UPGMA, neighbor joining, maximum parsimony and maximum likelihood. As you can see, we'll be using UPGMA for our project. So here we have used the muscle alignment, where we input file is sequence.fasta and output file is 1. We'll be using the muscle command line for this, and here as you can see the traits have been generated, and here a new FASTA file is created for the multiple sequence alignment. And here we create a phylogenetic tree using the bio.phyllo using biopython for it. Second, then using the phylogenetic tree, as you can see there are clusters formed for the common. These two W030, W145 fall under one cluster, C005, C084 fall under one cluster, W136, W219, C002, W077 they form under one cluster. So we will be using the phylogenetic tree to find the common ancestor. This means the the things in this cluster are closer in the evolutionary timeline. They are similar. So here we group them according to the clusters, like cluster 0 has 3, cluster 1 has 3, cluster 2 has 1. So these are in this list, all of them are similar and closer in the evolutionary timeline. Then we prepared a data set, like for C051, the sequence is this, subpopulation is this, subpopulation is intermediate, temperate japonica, and height is 86.17. Starting with the prediction using clusters by finding the most similar cluster. First we take an input sequence and then we find which gene it is similar to in our data. Then we send it to the cluster. Among that cluster, then we make it. Then here we built a website where this is the website where height prediction is done, where we select the phenotypic data, like this goes to height, and then we predict. After doing that, the prediction sequence is this. We enter a sequence, and after entering a sequence, it gives us the height. The predicted height of the tree is 120, 137 centimeter, which belongs to the subpopulation. And this is my project. Thank you. So this is my project for this. Thank you.